about using perfusion models as a platform to study the mechanisms and cell fate in target organs during normothermic machine perfusion. So I'll give you an overview at a high level of ex vivo normothermic machine perfusion, what it is, what it can do, what we can learn from it. And then I'll go through some case studies, starting off with using DNP, which is a drug in liver perfusion, modeling the effects of IL-1 beta on neutrophil infiltration in lungs, a project that we did with one of my students. And then looking at application of cellular therapy in kidneys, in that case, to try to get more organs transplanted. And then I'll open it up for any questions. So looking at ex vivo normothermic perfusion, or EVNP as it may be termed throughout the rest of this talk, essentially we take one or sometimes more organs, be it lungs, heart, liver, kidney, and we attach it to a circuit, and that circuit tends to contain a reservoir where we have the basic components of blood. Frequently, we may just use packed red cells and various other agents, pharmaceuticals to manage this so that we don't have the immune components there for a sort of clinical application of these. However, you can put complement agents in there or other circulating cells looking at neutrophils later. This then is drawn by a pump, and it goes through an oxygenator, a membrane oxygenator, which allows oxygen to enter into the blood cells, and a heater-cooler unit that then keeps it at 37 degrees. Frequently, there's a leukocyte filter. This can be circumvented. And then it goes past the temperature probe to maintain things through the organ and back through. And we do have a number of ports that are typically built into these circuits where we can then sample or infuse various agents including cellular therapies. And there's a variety of models we can look at. So we can look at isolated human organs without the rest of the system from organ donors. We can look at both healthy organs. These are organs that will have been declined for transplant, but perhaps the donor had some kind of a tumor that was found, and so they wouldn't want to risk putting them in, and we do then get organs like that. Or you can look for diseased organs, looking at fatty livers or other organs that may not be suitable for transplant, but may be ideal for some of the therapeutics that people on this call might be interested in testing. We can also look at paired comparisons to minimize the donor-to-donor variability, particularly for kidneys and lungs, where we can then split the lungs, and I'll show you a bit about that. And we can add various things. I mentioned this, but ranging from traditional pharmaceuticals all the way through to cellular and gene therapies that can then change the organs during this period, and we can study the impacts of these various agents as they're circulating within the perfusion circuit. And finally, we can monitor quite a few different things from these circuits. So we can look at various functional metrics. In the heart, it might be the contractile force. In the lung, you're looking at gas exchange. And then in the kidneys and the liver, you look at urine and bile respectively. And you can then look at the composition of those agents. We can look at other physiological measures, so temperature, flow, resistance. We can look at biological responses, and these can be broken down by compartment. So we can take samples of the blood or perfusate that's circulating around, but we can also take tissue biopsies from various locations and use a number of imaging modalities while we're doing this. And this can tell us something about the biodistribution of whatever's going in, as well as pharmacokinetics, if that's something that's relevant to the study that's happening. So the studies I'm going to show you in a series of cases are all from students in the lab. This one is looking at dinitrophenol treatment during normothermic machine perfusion of steatotic or fatty livers. And it'll be presented next month by Sam Tingle, who really led and delivered on this work. And so the aims of his study were to look at DNP delivery, to look at the potential for defatting the livers. But in this study in particular, it was looking at whether there was any toxicity from this, and then trying to figure out the pharmacokinetics and pharmacodynamics of this compound in the circuit, so we can then look at trying to keep a constant level. And here you can see that we've got a similar circuit to what I described, but we've got 6 livers that were declined by all centers due to steatosis or being fatty. And they were then exposed to 25 hours of normothermic machine perfusion with one group getting a bolus of DNP at 1 hour and the control not. And we can look here at the donor details. There were no statistical differences between groups. And here's some images of what the livers look like. So these are quite fatty livers that we're looking at in these perfusions. And so we started looking at toxicity at some of the physiological parameters I mentioned, seeing if you see reductions in flow throughout the organs. And we could see with the red, I believe, being the DNP group, that everything is roughly the same between groups over time. And then looking at measures of can they clear lactate or glucose that we measure with a blood gas machine. And we see that each of them was functional in clearing the lactate, and that they had similar profiles for pH and glucose throughout the duration of the perfusion, which was quite promising. Looking at measures of cellular death, we had looked at LDH and ALT in the liver, and we saw that these were similar, not statistically significantly different between the different groups over these time periods. And when we looked similarly at biopsies for histology, there were no differences in the 2 groups. And what Sam was then hoping to look at is the concentration of DNP over time to establish some pharmacokinetics. And we saw that it was first order with just the liver clearing this over time. And he was able to establish then a half-life of 7.7 hours with various parameters that then allowed him to reinfuse more DNP to maintain a constant dose with time in a prospective cohort that can be seen on the right bottom. Building on this, DNP causes an increase in metabolism, which should be reflected by oxygen consumption. And indeed, over the first few hours, we saw a statistically significant increase in oxygen consumption relative to the control. And this tapered down over time. And when you compared that then with the concentration that was circulating in the circuit, we then saw this very nice correlation between oxygen consumption and the concentration of the drug. And this gives us an idea that it is the drug causing this shift. I'll now talk to you about some of the work of Tom Pither that was presented at last year's British Transplant meeting, here focusing on the application of EVNP as an ex vivo model to observe the impact of IL-1 beta on neutrophil adhesion in lungs. And so to start off with, it's been established from a Newcastle-led clinical trial of ex vivo lung perfusion in the UK that IL-1 beta is a predictive marker of whether or not transplanted lungs will survive or not in this platform. And it's because it's a pro-inflammatory cytokine, and so it's indicative that you'll get this inflammation. And as we go through, you can look at, we know that neutrophils will be circulating once the lungs are transplanted into the body. They'll detect activated endothelium, begin to roll and arrest, and then exit into the tissue where they then release a variety of compounds, including neutrophil elastase, which is destructive and may then lead to a rejection event or lack of function in the transplant patient. So for this study, we were looking at a split lung model. So before, when we receive the lungs, what we do is we dissect them apart. We isolate then the airway and the vessels, which we then cannulate and attach to our circuits. In one circuit, we had a control, and in the other, we added some IL-1 beta to artificially elevate this. And here you can see some donor parameters below. We were then also able to take fresh donor blood, isolate some neutrophils, and label them with CFSE so that we can then track them and infuse them after a period of time in perfusion into the lungs and track where they go. And here you can see a multi-photon image of labeled neutrophils which had exited into the lung parenchyma. And in the bottom, you can see looking at flow cytometry and the levels of neutrophils that were circulating at a given time. And when we compared this we took a variety of samples to establish the kind of kinetics of where these neutrophils are going with time. We see that they quickly establish kind of a steady state where the control had a higher level of circulating neutrophils versus the IL-1 beta doped perfusion. And this is indicative that the neutrophils are then infiltrating into the tissue. And when we look at that a little bit shorter time frame, we see that this establishes within 1 to 2 minutes, so only a couple passes through the circuit where they then go in and we maintain these circulating levels. We can then filter this all out at the end to try to get the whole story. And we again were able to see that there were lower numbers of filtered cells in the perfusate reservoir with IL-1 beta versus control. And now looking at our final study, where Emily Thompson presented this at both the British Transplant and the ESOT, where she was quite successful with winning prizes on this work. And it's looking at novel delivery of cell therapies and normothermic machine perfusion to reduce ischemia, reperfusion injury, and kidney transplant. And this was using the multi-stem product, which I believe one of the panel members' companies makes. But this is a licensed product that's been used systemically, and here we're just infusing it straight into a kidney to see if we can't improve the status of the kidney prior to transplantation, enabling use of organs that otherwise may not be used. And what we were able to find is that when we added the MAPCs during NMP, they actually improved urinary output. And when we imaged using ultrasound, we were able to see that over time, these stimulated flow, particularly to the medulla, which otherwise was not receiving much flow, and we know can cause functional issues post-transplantation. And then this caused a variety of markers, both in the urine, where you saw NGAL, a marker of tubular injury, increase, as well as circulating factors, looking at pro-inflammatory, anti-inflammatory cytokines that suggests that this is inducing a more tolerogenic phenotype and making the kidney more receptive to surviving the transplant process. And we were also able to see where these MAPCs were going, both in the kidney through biopsy, as well as, and I'll show you a bit more about that. So looking at the MAPCs in the tissue, we're able to see that here they're labeled in a red dye, but we can image them in the vessels and the tissue. And particularly, we were able to see that these seem to home towards the peritubular capillaries in the medulla, which may then be stimulating the biological effect that I just mentioned. But they're also found in the glomerulus of these kidneys as well. And when we looked at what was circulating, what was quite interesting is that the circulating MAPCs, only a small proportion of them were viable, and that most of the ones that were circulating were dead. and that this was upscaled versus passenger leukocytes that were monitored. To bring it all together, we've been able to show that EVNP offers a focused platform to study therapeutics in healthy or diseased human donor organs. I hope I showed you some proof of concept data that EVNP can use to answer a variety of questions ranging from toxicity to pharmacokinetics, immune interactions, and biodistribution. And we see this as a platform where future studies can help elucidate safety and efficacy for a variety of therapies without exposing patients to risk. And that's a key point. I'd like to thank, within the Transplant and Regenerative Medicine Lab, there's a variety of clinical collaborators that I have. John Dart from the heart, Andrew Fisher from the lungs, Neil Sheeran and Colin Wilson on the kidney, Simi Ali. The technicians, really Lucy Bates and Marnie Brown are our key technicians leading on the perfusion. The donors, their families, and the specialist nurses, and here's some patients with some of our students and our funders. The NIHR supported a lot of this work, and ideally, you've not all fallen asleep on me, and I'm happy to take any questions right now.
Hi, Bill. Thanks so much for a great talk. I'll start off with the questions. I know we haven't met yet. I'm Sarah Bush. I've taken over for Tony Ting, kind of leading the project with Emily Thompson and the group that you described on the use of NMP and MAPC for kidney injury. And it's really a new indication for me, so a lot that I'm still kind of coming up to speed on, but of course, really interested in all the work that you showed. And you showed the labeled MAPC that were found, and I believe the paper described finding them in the cortex and in the medulla. I'm wondering if you or others have looked beyond that 6-hour time point that you mentioned in terms of the retention of the cells or related cells in the kidney. And I kind of asked that because I did some work in my early days when I was still hands-on as a scientist here at Athersys, looking in models of diabetes. And I recall looking in more chronic time points and seeing some really interesting observations. So I'll let you answer first, and then I'll come back with more comments.
So I think that particular study was quite focused with answering the questions that we presented, and Emily wanted to do that as part of her fellowship. And so I don't think we've gone beyond. However, we could look at extending the duration of perfusion towards answering targeted questions, and perhaps we could have another call with her and Colin and Neil at another point down the road.
Yeah, that'd be fantastic. But I just wanted to mention that my observation, at least in a diabetic kidney, was that I was looking for the retention of cells. I believe it was potentially 7 days. I'll have to look back at that before we talk next. But I was looking at human mitochondrial antibody just as a marker and a way to track the cells in a rat kidney, and I found brilliant signal in the glomeruli of the kidney. And when I went back to verify that those were indeed cells, I used, I believe, 2 other methods and could not confirm that there was human DNA there aside from the actual mitochondria. So it looked that we were retaining mitochondria for a very long period of time once the cells themselves were actually cleared. So I'm just kind of curious, and I'll have to do some more digging and look back at that data to see if that might be a potential mechanism by which the cells can continue to exert benefit after they're gone.
Yeah, that's really interesting and something we could look at studying. I know Colin has been talking with me about extending the perfusion time out to, say, a week. However, I don't know that we've been doing that as of yet to date. So it would need to have a good reason, but we could then look at studying that, collecting biopsies, and I believe there's some of the new donor-specific DNA assays that you might then be able to look at the differences between the donor and the cell DNA source to kind of back some of that out, or looking at the mitochondria.
Yeah, so, that'd be really interesting. Great, thank you.
Thank you. Any other questions?
There's a comment on the chat box from Aaron Boswell saying that's a very nice talk. We have some more time for additional questions. Feel free, do not hesitate to either take the microphone or put your question in the chat box. Maybe I'll take one myself. In regards to the last part of your talk and the use of these models with cellular therapies, are there any other studies that you are thinking about or would like to take on?
Yeah, so we have some other students working on other cell types in different model organs. And so we're looking at human amnion epithelial cells as one in lungs, but that data I think is just being summarized now by our student Chelsea Griffiths. And certainly there are other cell types that we could be keen to look at either to benefit the organs, as is the case with the multi-stem, or it could be useful to look at how they might interact with various disease states to see what's happening with the cells over this initial period in these platforms. So happy for anyone to reach out if they're feeling shy, and we can have a discussion later. And otherwise, I guess I can let you get back to the rest of the meeting, Lucilia.
Absolutely. Thank you.
This is Aaron. I have a question, actually.
Yeah.
Hey, Bill. I'm new to the cell therapy world, and this seems like a very useful way to understand where allogeneic cell therapies traffic after infusion. How does this method fit into the kind of context of existing model systems for evaluating this? You know, would it be used in a preclinical setting, or I guess how do you envision it being implemented?
So I think that this could be a preclinical study at first, where you could take it as a human organ or multi-organ. We've done kidney and liver at the same time. You could then study in this isolated platform how it's interacting with human tissue or diseased human tissue before you then expose it to live patients as a final kind of go no go, where it's legit organs as opposed to cells in a dish or spheroids or something like that. Alternately, it does offer a platform for therapeutic delivery for transplantation and looking at benefiting these patients, because we get these because these are organs that are thought not to be usable in our patients. And so we're trying to then take these and make them usable so we can save more lives, as there's large waiting lists, as you know, or you may be aware of patients who unfortunately will die if they don't receive this kind of therapeutic.
That makes sense. How available are these organs if people wanted to implement, to use this as that no go kind of state before putting in humans? Is there a waiting list for rejected organs as well as viable organs?
So in the UK, we have a system where they'll take the organs for us. We get what's called our research advisory group approval and a ranking, and then we can take these in. It depends on what you're looking to do. I can say that there's a large amount of, say, fatty livers that are discarded every year that we could then use to study various effects. And NAFLD is quite a large problem facing the world at the moment. And other organs. But for other organs, we've been taking steps to unlock this potential. We just launched an initiative called Increasing Numbers of Organs for Research, imaginatively. But theoretically, that should provide a large number of lungs, hearts, and could impact other models. So it depends on the organ. Kidneys tend to be used the most because they're more robust in general. But then there are a variety of organ models that we could get, and it's a, what do you call it, chicken and egg issue. If we have demand for more organs, then we can go back to NHSBT and here in the UK and kind of ask that we need these tissues. Otherwise, if they're not being utilized, there's less impetus to remove them and ship them around with the associated costs.
Right. Has anyone done this in brain?
I don't know about the brain. I think there was talk of someone doing a head transplant a few years back. There was a billionaire who was paralyzed from the neck down, who had to deal with China and Russia to do something. I don't know terribly much about that. I think that his wife may have convinced him not to proceed with that route. But in the UK, everything that we use is brain dead by law. So we determine death by brain death. And so I don't think we could get brains for you. In animal models, you may be able to make a justifiable case to the ethics board to then study this, but that's a separate talk maybe down the road.
I hadn't really thought through that question about brain. It makes sense. Thank you.
No worries.
And there are a couple more questions in the chat box from Martha Lundberg. Would you like to take those on, Martha? Do you want to?
Sure.
Go ahead, please.
Can you hear me?
Yes.
Yeah.
Okay. Yes, the great talk. I really enjoyed your data, and you know, you've been really using your students' strengths in your lab, so that's awesome. So I am at the National Heart, Lung, and Blood Institute, and one thing that, you know, as you obviously know, is that, you know, the transplantation time for hearts is very limited. I mean, 4 to 6 hours is maximum. I was wondering if you have looked at any data, or have you done any work to show that allogeneic cells could prolong the viability of the heart? And with that, also I had a question is, you know, the tissue preservation technologies that are currently under development, could those affect the cells that you might use to show improved viability of hearts?
So thank you for those nice questions and the praise. So for heart transplant, at the moment, we do have models where we can then study either hypothermic perfusion in the heart or normothermic reperfusion of the heart, and then looking at a variety of agents. I didn't show the data that was sent in just last night from John Dark, who leads on the kind of heart perfusion side, but certainly it offers a platform to look at damaged hearts. There's a huge cohort of circulatory death or cardiac death donors. So about 40% of our donors in the UK come from that pathway. And that's where they then withdraw care and wait for you to have a cessation of heartbeat for 5 minutes, where we could look at that. Or we could intentionally induce some damage to the heart on the platform to then look at the impact of cells in that. We've not done that yet, but there are plans in the works to start looking at things in the heart over the next years. In terms of how the preservation technologies affect the cells, what you can do is model that without the organ present to try to then dissociate the two and see that they're not being impacted by the shear flow of the circuit. In the circuits that we use, it has a centrifugal pump, which minimizes kind of the direct compressive impact on the cells. But we can then study if they're then binding to tubing in the circuit or what's happening to them independently. And then when we add the tissue to the system, see the impact dissociated from the impact of the circuit itself. I hope that made sense.
Yes, it does, and you know, I think this is a very understudied area and an area of great need. So anything that you can do to, you know, improve viability for hearts, that would be so great, because you know, as you know, the heart is like the most sought after organ, but so few people can get them. So awesome job. Thank you, Bill, for all your hard work in this area of transplant and allogeneic cells. Yep.
Bill, I just wanted to mention real quickly that Athersys is also really interested in the heart work. I'm not sure if you were aware of the work we previously did with Mark Dahlke. That was more focused on looking at immune modulation in a heterotypic heart transplant model. I'll find the reference and put it in the chat. But I think it's something that ties really well together with the rest of the work that you described, and we're very interested in pursuing that as well.
Yeah,
awesome.
Thank you.
All right. Well, thanks all for the questions. Are there any last comments or question for Bill before we wrap up and move to the next agenda items? And I see that Sarah just put the link of the study she mentioned in the chat box. If you're interested, the link is there. Thanks, Sarah.
Thank you.
So if there aren't any further questions, I'll thank Bill again for your presentations and everyone for participating in the questions and discussions. Very interesting work indeed. If you have further questions, feel free to follow up via email, and we can come back to it at the next meeting if needed.